Dear viewers, greetings. In this video, we are going to see about the electron microscope and its types, transmission electron microscope and the scanning electron microscope. First, the electron microscope. The electron microscope was first developed by the scientist Ernest Rutherford and Max Knoll in the year 1931, and the electron microscope magnifies the objects from 10,000 times to 1 lakh times through the millions of times magnification with good resolution. And the electron microscope is used for observing the objects which are smaller than 0.2 micrometer, such as the viruses or the internal structure of the cells. In electron microscopy, a beam of electron is used instead of the light. Usually for the light microscopes, the light can act as a light source, but in electron microscope, a beam of electron can be used instead of the light. And the resolving power of the electron microscope is far greater than that of other microscopes like light microscopes. And instead of using glass lenses, the electron microscope uses the electromagnetic lenses like condenser lens, objective lens, or projector or ocular lens. There's three types of electromagnetic lenses are present in the electron microscope. And the electron microscope uses electromagnetic lenses to focus a beam of electrons onto the specimen. In electron microscope, the produced images are black and white, but they may be colored artificially to accentuate certain details. Then basically, the image produced by the electron microscope is black and white. Next, the photographs taken on an electron microscope are called as electron micrographs, and it provides the detailed view of smallest bacteria, viruses, internal cell structures like cellular organelles, biopsy samples, metals, crystals, large molecules, and large atoms. The cellular structures that can be seen only by using electron microscope are referred as the cell's ultrastructure. We are studying the ultrastructure of various microbes like bacteria, fungi, algae, protozoa, and viruses. All the ultrastructures are studied with the help of the electron microscope. Regarding the parts of electron microscope, the electron microscope contains four parts. One is the first part is the electron gun. The electron gun is a heated tungsten filament which generates the electron. Usually in the light microscope, the light source, any mercury, any lamps will generate the light. Here there is no lamps. Here the electron gun is present. This heated tungsten filament present in the electron gun generates the electrons. The second source is the second part is the electromagnetic lenses. Three types of electromagnetic lenses are present in this electron microscope. First one is the condenser lens. Here, two condenser lenses are present. In the normal light microscope, only one condenser lens is present, but in electron microscopes, two condenser lenses are present. The condenser lenses focuses the electron beam onto the specimen. That is, the first condenser lens focuses the electron beam to the specimen, and a second condenser lens forms the electrons into a thin, tight beam. Usually the main role of the condenser lenses is condensing the things, and here the electrons emerged from the electron gun are condensed with the help of the condenser lens. The second one is the objective lens. The electron beam coming from the specimen passes down the second of magnetic coils, that is called as the objective lens. The objective lens has a high power and forms the intermediate magnified image. The third one is the specimen holder. The specimen holder is an extremely thin film of carbon or collodion held by the metal grid. And the fourth part is the image viewing and image recording system. The final image is projected on the screen. and that screen may be fluorescence screen for transmission electron microscope. And in scanning electron microscope, the final image is projected on the PC screen. And below the screen, a camera for recording or image will be present. These are all the 4 important parts of the electron microscope. This image shows the scanning electron microscope and the transmission electron microscope. The first type of the electron microscope is the scanning electron microscope. The first scanning electron microscope, in short term it is referred as SIM, was debuted in the year 1942 with the first commercial instrument around the 1965, and its late development was due to the electronics involved in the scanning the beam of electrons across the sample. And next, working principle of scanning electron microscope. And here in scanning electron microscope, the specimen is coated with a thin film of the heavy metal. Typically the gold is the heavy metal used for this. The specimen to be examined is made extremely thin, that is at least 200 times thinner than those used in the optical light microscopes like phase contrast fluorescence, bright field and dark field. And in scanning electron microscope, first the electron source. Here in this, an electron gun produces a finely focused beam of electrons called as the primary electron beam. Here the primary electron beam is produced, and the primary electron beam passes through the 2 electromagnetic condenser lenses. Here 2 condenser lenses are present. This is condenser lens 1, this is condenser lens 2. The formed electromagnetic beam will pass through these 2 condenser lenses, condenser lens 1 and condenser lens 2. Okay, the first electromagnetic condenser lens, this lens, the first electromagnetic condenser lens largely determines the spot size and the general size range of the final spot that strike the samples. Here it first determines the spot size, and the second electromagnetic condenser lens, this is the second electromagnetic condenser lens. This second electromagnetic condenser lens changes the size of the spot on the sample and changing it from a wide dispersed spot to a pinpoint beam. The second condenser lens converts the wide electronic beam into a pinpoint beam. See the difference here. The electron beam is in wide. Here it is changed as the pinpoint beam. And next, the beam from the 2 electromagnetic condenser will direct over to the surface of the sample through the parts like aperture, scanning coil, objective lens and backscattered electron detectors. And after traveling, the primary electron beam knocks the surface of the sample, and here the interaction occurs with the sample and produce the secondary electron beam. Here these 4 arrows formed represents the secondary electron beam. The produced secondary electron beam are transmitted to the detector. This 4 arrow, that is the 4 blue arrows, shows the secondary electron beams which are produced by striking of the primary electron beam into the sample. The interaction between the sample and the primary electron beam will result in the formation of the secondary electron beam. The produced secondary electron beam are transmitted to the detector. This is the detector is present here. This is the detector, and the detector, from the detector it is moved to the amplifier and used to produce an imaging on the viewing screen on the photographic plate. Here in the PC screen the image will be observed. Okay, the image which are observing on the screen is called as a scanning electron micrograph. Okay, first electron source produce the electron beams, that is the primary electron beams. That primary electron beam will pass through the two condenser lenses. Here the condenser lens make the electron beam as a pinpoint. The formed primary electron beam pass to the sample through the aperture, scanning coil and objective lenses, and after the interaction with the sample, the primary electronic beam will produce the secondary electron beam. The produced secondary electron beam move to the detector and then move to the amplifier and amplify, and finally it produces the image on the screen. This is called as scanning electron micrograph. And this image shows the gram-positive bacterial cocci under the scanning electron microscope. This image shows the gram-positive bacterial rods under the scanning electron microscope. This image shows the gram-negative bacterial rods under the scanning electron microscope. This image shows the fungal mold under scanning electron microscope. And this shows the algae under the scanning electron microscope. And this shows the amoeba under scanning electron microscope. This shows the ciliated protozoa under scanning electron microscopes. And this shows the Ebola virus under scanning electron microscope. Regarding the uses of scanning electron microscope, the SEM is very useful in studying the surface morphology of the specimens, macromolecular aggregations and tissues. It can able to magnify up to 1000 times to 1 lakh times. And the SEM has a variety of applications in a number of scientific research and industry related fields. The second type of microscope is transmission electron microscope. This is the first type of electron microscope to be developed and is patterned exactly on the light transmission microscope, except that a focused beam of electron is used instead of light to see through the specimen. This transmission electron microscope was first developed by Max Knoll and Ernst Ruska in the year 1931. Both Max Knoll and Ernst Ruska are the German scientists. Regarding the working principle of the transmission electron microscope, this transmission electron microscope requires a vacuum system consisting of pumps and their associated gauges and valves, and power supplies are also required for this transmission electron microscope. And here in the scanning electron microscope, the sample is present in the end, but here the sample is present in the middle. The specimen or sample to be examined is made extremely thinner than those used in the optical light microscope and the scanning electron microscope. The specimen here which was visualized here should be very thin than the scanning electron microscope. Typically the ultra thin sections of 20 to 100 nanometer are cut, which is already placed on the specimen holder. The sample will be placed in the specimen holder, and here are like same here also a finely focused beam of electrons from the electron gun passes through the specially prepared ultra thin sections of the specimen through the electromagnetic condenser. The electron beam formed from the electron sources pass through the two condenser lenses. One is the first condenser lens, second one is the second condenser lens. And here the first electromagnetic condenser lens largely determines the spot size and the general size range of the final spot that strike the sample. And the second electromagnetic condenser lens changes the size of the spot on the sample and changing it from a wide dispersed spot to a pinpoint beam. The wide part of the electron will be converted into the pinpoint beam here. Next, the sample is usually placed in the placed on a copper mesh grid, and the beam of electron passes through the specimen and then through the electromagnetic objective lenses, and here the beam of electron passes through the sample and then through the electromagnetic objective lens, and here the electromagnetic objective lens magnifies the image, and from the electromagnetic objective lens the electron beam will pass through the object aperture, and the object aperture enhance the contrast of the image by blocking out the high angle diffracted electrons. Next, from the objective aperture, it passes to the selected area aperture. The area aperture enables the user to examine the periodic diffraction of electrons by orders or arrangements of atoms in the samples. And then the sample is passed down to the intermediate lenses and the projective lenses, and these both lenses are being enlarged the image all the way, and finally the electrons are focused on the fluorescence screen, which is also called as the photographic plate. The final image formed in the fluorescence screen, and the final image formed in the fluorescence screen or the photographic plate is also called as the transmission electron micrograph. That's also here the main difference is in the SEM the sample is present in the end of the session, but in the TEM the sample is present in the middle. And below the sample, objective lenses, objective aperture, selected area aperture, intermediate lens and projective lens are present. That's all about the working principle of the transmission electron microscope. This image shows the bacterial cocci and the transmission electron microscope. This figure shows the bacterial rods under transmission electron microscope. And this shows the fungal yeast under the transmission electron microscope. And this shows ALGAE CHLAMYDOMONAS under transmission electron microscope. This shows the VACCINIA virus under transmission electron microscope. And this shows the BACTERIOPHAGES under transmission electron microscope. And uses the TEM is used to show the ultrastructure of the cell, crystallization, viruses and tissues. Mostly the viruses are observed using the transmission electron microscope. In the scanning electron microscope, the electrons scans the surface of the cells, but in transmission electron microscope, the electrons will pass into the cell. So it is very helpful to study the ultrastructure of the cells. And the TEM is very ideal for different fields such as the life sciences, nanotechnology, medical, biological, and material research, forensic analysis and metallurgy. It also provides the morphological, compositional, topographical and crystalline information. And in general, the scanning electron microscope scans the surface of the object, but the transmission electron microscope will passes the electron beam within the object and it forms the ultra clear images. That's the benefits. Regarding the advantages of electron microscope, very high magnification, incredibly high resolution, materials rarely distorted by preparation. It is used to investigate a greater depth of fields, and it also have the diverse applications. Next, regarding the limitations, more expensive than the light microscopes, it takes much more space. It requires highly skilled persons to operate. The live specimens cannot be observed in the electron microscope. As per the penetration power of the electron beam is very low. The object should be ultra thin. For this, the specimen is dried and cut into ultra thin sections before the observation. The thick smears will not be allowed to observe under the electron microscope. As the electron microscope works in a vacuum, the specimen should be completely dry. That's all about the electron microscopes. In the next video, we will see about the difference between the scanning electron microscope and the transmission electron microscope. Thank you for your support. Thank you.